All right, I'm gonna welcome everyone here to I don't know if I told you, Brandon, yours is the first talk of this quarter, of winter quarter, in this Wednesday at NICO seminar series. And we have a really interesting speaker for you all today, Brandon Ogbunu, from visiting us from Yale. I think he's got an amazingly varied background, interesting background scientifically. So your undergraduate degree was in chemistry, as I read, but Brendan has moved into what I would call, he's moved into evolutionary biology, but also complex systems. So after Howard, I think for your undergraduate degree, he was a Fulbright scholar, something we share actually. Wish we had more time to chat, and then went to Yale for his PhD. He was a postdoc at Harvard, and then faculty at University of Vermont, which has another great complex systems program, before moving to Brown and then on to Yale, where he is right now. But during this time, he's also had an affiliation with the Santa Fe Institute, another amazing hub for complex systems. And he is a resident faculty right now there, in addition to being faculty at Yale. And I missed the visiting faculty at MIT also. So Brandon's been everywhere, and all the best places in particular for complex systems. He also writes popular science articles that you may have seen in the media and Wired and The Atlantic. His research, I would say, focuses on evolutionary biology, my take on his research, but also connects to culture as well. And I'll throw in one last tidbit I read about from an interview published with you, that you're on the board of a theater collective, I believe also, or you were, which is another amazing intersection with culture. Anyway, I think the science is great. Brandon's a really interesting person, and I'm excited to hear his talk today. So let's welcome him.
Thank you. Thank you. Really, really excited to be here. Thank you all. I'm happy to kind of lead off for this season. You have some remarkable speakers, so I'll set the bar nice and low so the rest of them can come in and do their talk. No, I'm just kidding. I think I remarked this morning about the conversations I had. We talked about the circadian rhythm, and then we talked about kind of information spaces and scaling laws, and then we talked about robotics, and I've been back to back, and that's the type of kind of engagement I don't get, certainly not on my day job at Yale. So that's a testament to the sort of range and depth that exists at this institution. So congratulations to the leadership and all the trainees and all the faculty and administrators for making this place kind of rock that way, because I think it's very, very exciting for somebody like me. This title, Soft Rules for Biological Engineering, Context, Complexity, and Evolutionary Genetics and Beyond. You heard it in introduction. That was very kind of you. Thank you. And we're going to kind of talk about various things. Now, I think the most important kind of component here, as we talk about a talk, is I have a very particular style of doing a talk. I think the scientific seminar is one of these dimensions that really needs disruption. I'm one of those people. In the sense of, I think seminars are best when you're making an argument about the manner that you look at nature, and you use your work as a way to communicate the way that you look at nature. It's not just like your CV, okay? And like the papers you've published. I don't care, to be honest with you. I can read those. I want to hear kind of what you have to say. That said, you're going to get a kind of a library of published work and some of the, you're going to get this and other stuff that we've done in various combinations. And obviously, I'm happy to send these things to you. So you're going to get them. The reason why I have a tape here is this is my generation. I look at talks as kind of like mixtapes of your kind of science. You gotta like take a song here and take a song here, and you put them together, and you're able to kind of tell a different story about kind of the way you look at the world. So that's kind of how we're going to do things today. And fundamentally, we'll ask this question. We'll examine fundamental concepts that complexify our quest to manipulate biological systems using genetic and genomic interventions. That's going to be the key thing that we talk about here as my model problem, something that we're working on. Before diving in, kind of in many ways, the most important part of a seminar is going to be kind of the people who have helped make this happen. This is my research group at Yale, and you'll meet some of them along the way, but really, really terrific. I've been able to build out the sort of culture that I want within my laboratory, which is very, very exciting. all of the funding and administrative support from various institutions. I think these ideas go back to graduate school and have been reanimated at various points in my career. Very, very grateful to them, all the administrators who make this possible in various ways. All of the former mentees who are now mentors, former mentors who are more mentors. I think they kind of contributed to this kind of emergent phenomenon, which is my research interest, and I'm very grateful to them for this. And then my newest scientific family, the Santa Fe Institute, and I have several jokes about the place, but you ever feel at a place your whole life? Anybody? Right? And then you go to a place and everybody's stranger than you are. That's what the Santa Fe Institute's been for me. And I think it's kind of helped empower kind of the way that I think about nature. So I thank them for that. Now, we heard this great introduction, and I like how you kind of phrase this. You're like, to me, from my perspective, you're an evolutionary biologist, which is a good way to think about it because I'm kind of whoever you think I am. Is kind of thinking about things. But I think there's some ways we can kind of contextualize who I am. You know, computational biology, evolutionary systems biology, I've been called, disease evolution in college, I've been called an epidemiologist or a complexity scientist. But fundamentally, my group uses diseases as systems to ask questions about evolution, complexity, bioengineering, technology, culture. And we invoke the study of evolution, complexity, bioengineering, which you'll hear about today, technology and culture, to understand diseases and kind of other social phenomenon as well. So that's kind of one way to describe it, you know, on a website. And I think the heuristic that we use is this. This is really kind of fundamentally where I live. The context shape biological systems, no argument there. I think everyone would probably agree who thinks about biological systems. They shape molecular evolution, they shape how epidemics manifest, and they shape science and the world in which it's all embedded. Right? This is kind of how I think about this. Right? What are the actors that drive complex biological systems? What is the nature of those interactions in the context that modulate these? So when we think about what context is, oftentimes we hear that, particularly these days, when you hear the term context, it's kind of like it's a politician or somebody like that, or a celebrity who says, oh, no, that was taken out of context. Or maybe we've had a text message exchange with someone and we were like, well, text isn't the really right way for me to communicate this. Or we say because of social media timelines being short that, right? So context is like this thing that we hear very, very commonly, but I think it manifests in the sorts of biological, and really in all of our work. I can, you know, we can go around and we just had this beautiful conversation about the circadian rhythm and the sorts of inputs that influence kind of how a fly constructs its idea of kind of the time that it's in, as an example. But I think one way I think about it, if you're in ecology or maybe you're in quantitative genetics, this is all called the norm of reaction. This is an abstraction in ecology and quantitative genetics where you have just a trait of some, could be how tall a tree grows, and this could be temperature, this could be some kind of geographical. And it basically just says that, right, it anchors a lot of the ways we think about quantitative genetics. In this case, there's no real biology here. It's just you're interested in context and how it's changing the you know, some kind of trait value. We could think about it if you think about proteins at the molecular level, Professor Braun, for example. We could think about like how temperature is influencing how an enzyme's functioning. Okay, that maybe is a microenvironment, and here's a microenvironment that's influencing the way an enzymatic process is happening. We'll meet this enzyme later. This is dihydrofolate reductase, but I think my group thinks quite a bit about this. If you think about ecosystems ecology or conservation biology, maybe you're thinking about context this way, okay? I have a graduate student who's doing beautiful work on kind of historical epidemics, right? And kind of mathematizing and kind of recapitulating the sorts of forces that craft how diseases became endemic and travel across the world. So these are historical forces, right? These are wars and the role they play in epidemic dynamics. So that's a sort of context that we think about. And then there's context like this. I'm in, you know, just outside the great Chicago. Any New Yorkers in the room by any chance? There's often one. How about that? Look at that, right? Okay, we got one. So I'm not gonna ask you to identify this. This is the Queensbridge houses. Actually, Chicago's quite relevant because Queensbridge Houses is the largest public housing complex in the United States now that the Robert Taylor homes in Chicago was actually torn down quite a bit ago. All right, and the idea here is in musicology, for example, there's a great question of why it is that this neighborhood gave birth to some of the greatest wordsmiths to ever exist. And these are hip hop artists in the 1990s, many of them my heroes. But if you look at this photo, right, there's actually biology going on. There's like a tree growing, right? And as you know, urban ecology and evolution is a real field now. How do urban environments craft the environment that natural selection is kind of operating on? And so when I think about my work, right, we think about kind of how context is framing biological systems, and depending on your question, right, these various dimensions of context are kind of influencing the system, right? In terms of epidemiology, we've learned how all of these sorts of different contexts can influence disease systems. So right? We asked this question about context environments and how they shape. And so there's three kind of pillars to my research program. What context shape the evolvability of pathogens? So I think about that, and I think we're, right? And I think on a scale up, we think about what context shaped the impact of epidemics. And then there's another scale up, and some of this you heard about, right? With social context shape epidemics, social institutions, culture, and science itself. And I think this, and many of the best people in the world here, it's particularly on the science side, kind of live in this department. So I think you all are familiar with this. And I think we're able to animate these things. And I think I have talks I can give on all three. I have talks I can give on all three. I can give individual talks about each. I think today, because we've been thinking about kind of the deeply molecular, we're going to focus on this molecular dimension to the work, just because we've been having some fun recently thinking about this problem and the manner that it is becoming a frontier of complexity science as I see it. So how do the laws of evolutionary and population genetics inform contemporary efforts in biological engineering is going to be the question here, right? So there's at this molecular level, there's fundamental principles that have existed for quite some time, but are they relevant, and how are they relevant for the sorts of challenges that we're thinking about in biological engineering and specifically genome engineering? Now, to animate this question, we're going to kind of use a model problem, a model system. I think many of you have them. This probably isn't a too model system heavy. I think we talk scaling laws as like a tool that you use to look at the universe, for example. I think there's other sorts of things. I think we'll start with a kind of a very basic question. What is a mutation and what does it do? You can boil down a lot of the things that I'm interested in to kind of what is a mutation and what does it actually do? There's a Wikipedia definition, an alteration in the nucleic acid sequence of the genome. That seems about right. I think that probably matches what our idea is. Of course, this isn't where I learned it. I didn't learn it from population genetics, and I didn't learn it from Wikipedia. I actually learned it from here growing up. Okay. Some of it they actually got right, believe it or not. Like X-Men is deeply about, right, the context dependence of a mutation, and the reason why you have different powers is because of the context dependence of a single mutation on the chromosome, on the X chromosome, hence X-Men, right? And Ninja Turtles less so, but also quite entertaining. But the idea here is we can think about this question about what a mutation is doing is foundational in all of biology, and certainly in the various subfields of genetics. Right, you swap something, what does it actually do? We had a conversation earlier with Todd about there's this path dependence that starts with Gregor Mendel, where we have a deeply simplistic understanding of what a mutation does. Right, you have a mutation does here, you get these certain sorts of phenotypes, and I think this has kind of driven the field with regards to the manner that we study genetic information and alterations in genetic information. Right, but there are other framings we can think about. Other applications. We can ask this more fundamentally, right? In population genetics, for some of you, you might understand that we study this in kind of with some pretty formal statistical genetics and population and evolutionary genetics and quantitative genetics. This thing called the distribution of fitness effects. If you take a gene, what is you can basically put their effects. Is it beneficial, neutral, or beneficial based on some kind of distribution? Certain genes have different genes, and different organisms have different. And we think of some fundamental property of genes and genomes. I think about this problem, but I think about it much more on the physiological level. What is an individual mutation actually doing to a gene or protein? But I think we thought about this quite a bit here, of course. Remember these narratives for how individual mutations that were in the news, right? And what are they actually doing to the pandemic? And we have E48K, that's EEK. And then D1640 was DIG. And, is there a DIG mutation? And this is actually part of the molecular narrative of the pandemic, was us learning and then kind of unlearning that the story isn't that simple. This was actually kind of part of the narrative there. And these were essentially very, very important, particularly for epidemiological tracking. But in terms of function, it's actually a little bit dodgier than that. And some of that we'll get into. So the idea here is that even a mutation itself, the relationship between genotype and phenotype, when you swap a gene in, you drop a mutation in, you engineer certain things, we have expectations for the manner that it's supposed to happen based on a relatively simplistic picture, right, of how information flow happens in biological systems. And this is not wrong, right? Like a lot of complexity science, it's not that the simple thing is wrong, it's just ain't enough. Right? And that's kind of going to be a theme here. How can we actually imbue our perspective on this with a little more jazz? We'll employ a kind of a model system for this, and that will be the fitness or adaptive landscape. Some people from computer science know this. Linguistics has their iteration. There's a lot of different people who think about it. And basically, it's just a relationship between genotype and phenotype that kind of likens evolution to like climbing, like, right, some kind of physical surface where you're moving uphill and downhill. This has been a transformative analogy in evolutionary biology. And it was kind of introduced by Sewell Wright, kind of luminary of the modern synthesis of evolution, right? His main area of intrigue involved moving from peak to peak. How does evolution locate solutions to complicated problems in this kind of terrain, this complex terrain, where some mutations are taking you downhill, some of them are taking you uphill, and what have you. And you see a representation of genotypic space where populations were clustered, could move from peak to peak via the various forces. It could be migration, could be mutation. So this has kind of been a foundational kind of analogy that, again, has been applied across various domains. This will be the thing that we use to talk about these problems today. And the molecule that we use is dihydrofolate reductase, which is an essential enzyme that is highly conserved. And the reason why, for that reason, it's the target of antifolate drugs. Everything from antibiotics to antimalarials to anti-cancer drugs or antifolates. It's a class of drug that actually binds a pocket of DHFR. It's remarkably well conserved. And so because of that, this is one of the better studied proteins on Earth. And so we'll kind of use this as our model protein. Every project you see, I think, will be in dihydrofolate reductase in various species. So where are we now with regards to biological engineering and genome engineering? Why is this an important frontier for like the complexity science framework and the way that I think about things today? Well, there's some kind of fun examples, and then we can kind of summarize where we are in some ways. I think, I argue we're kind of at the end of the age of genomics, where we were just like, wow, we can sequence things, we can sequence things. Now I would argue we're in the post-genomic age, where it's like, okay, what do we do? Right? And now what do we do, right? So we have the tools to uncover the genetic basis of phenotypes. It's amazing. But not only that, we now have the tools to modulate information and tinker with it in a very, very rigorous way. And so we can think about some notable kind of examples to facilitate our conversation about where we are and how complexity science can help us think about the bioengineering dimension in particular. This was 35 years ago. All right. Okay. So I have a technical paper now in press, which we'll hear about, right, where I actually use this anniversary to talk about contemporary issues in genome engineering, right, in a new theoretical biology journal. But we don't have to use this because this has been invoked recently as an analogy or a comparison for something that's actually not fictional, but very, very real. Let me give you a hint. Okay. That's actually J.R.R. Martin. I saw him at the movies in Santa Fe. It's very cool, yeah. It's about, right, you might have heard about the de-extinction of the dire wolf, okay? Now, the dire wolf, of course, is famous from Game of Thrones. There's an ethical conversation and a philosophical conversation I'm not gonna have here. I think it'll kind of manifest in this conversation in some ways. We're gonna try to keep it technical. We're gonna try and keep it about the technical issues involved in de-extinction. And basically cover the cover of Time magazine. Allegedly, we actually were able to de-extinct an extinct animal. That's what don't ask me, look at the Time magazine, okay? Right? And this was a really, really exciting thing, but when you dig a little bit, right, look at this. I mean, this is adorable. Look at this. I mean, dire wolves reborn. This is great. And we were able to presumably take an extinct animal and bring it back. That's the way it was reported. I see some heads shaking right here. I'm with you, brother. We'll get there. We'll get there. Right? This was from the CEO. The massive milestone is the first of many coming examples demonstrating that our end-to-end de-extinction technology stack works. And $10 billion of funding, and Tom Brady's an investor, and George R.R. Martin is an investor. And it was this whole thing about our ability to kind of bring species that are back. Okay? So we'll talk about this a little bit more carefully in the sense of this is just kind of one example of all of these contemporary sorts of debates and controversies where we're living, where the science of biological engineering and genome engineering is now kind of has been controversial. So this is like the tip of the iceberg. It's the latest, right? Genetic discrimination isn't in theory. It's already happened, okay? The controversies around gene editing, right, confusion over kind of the genetic bases of complex diseases, of course, the genetically modified crops debate. I mean, there's been a lot of conversation about how do you actually modulate biological information in the genome, and should we be doing it? But again, the ethical part, which will live in the background of this conversation, I won't have now. We can kind of talk about that over lunch or whatever. But more than that, we'll talk about some of the technical misgivings. Right? What are some of the things that we're actually missing from a technical dimension that classical evolutionary and population genetics might be able to help us with? And this is real. This is not my complaint. Don't ask me. Okay? I mean, these are real articles, right? The companies that are actually trying to use CRISPR-Cas to change genetic modifier, I think, are failing. Flat out. Not my opinion. Okay? And so the idea is, why are they failing? It could be because of mismanagement or the lunch breaks are too long, but it's probably because there is something technical being missed about the central problem of modifying a genome and expecting it to do something, okay? I think something analogous is happening in terms of microbiome therapy, where the challenges are at least analogous, if not the same, and we could talk about that. So that's kind of where we are with things. And so we'll introduce the extinction in genome engineering, some of the things that are news, and we'll introduce 3 concepts that are going to form the central part of this talk: plasticity, pleiotropy, and epistasis. And we'll explain and define, and I'll highlight for you how they're relevant, and then we'll summarize with some soft rules, as I call them, for biological engineering. So let's talk about the science of dire-wolf de-extinction briefly, just because it will kind of reveal some of the challenges here. So they recovered a full dire-wolf genome from a tooth. I mean, that's awesome. I'm sorry. That's cool. The fact that you can recover a genome from a tooth, I mean, can we live? Okay, it's just some squinting here. Some people aren't buying it. I'll stand by that. It is very, very fascinating. There's a great book by Beth Shapiro about this called How to Make a Woolly Mammoth or something like that. It's really, really quite good. So there's a really, really sophisticated ancient DNA technology that we should applaud. But what they, right, and so use multiplex CRISPR-Cas technology. 20 mutations were engineered into the gray wolf, which is here, across 14 genes. So really what they did, and listen, we're not being cynical. What they did is they engineered specific mutations from the dire wolf, the extinct dire wolf, into basically a modern gray wolf. Made it look like a dire wolf because of the physical traits, and basically said, we resurrected the dire wolf from extinction. So by no definition is that a de-extinction event, and we don't need to argue about that. I'm just talking about what actually happened at the molecular level. But there's some other interesting things. It was 20 mutations in total. 15 were from the extinct dire wolf, which is cool, right? But 5 were variant alleles of the gray wolf, right? Why couldn't they engineer, and stay with me here and focus on this. Why couldn't they use all the mutations from the extinct dire wolf? They couldn't use them because some of them caused, right, unforeseen consequences when you drop them into a new organism. And so they had to use 5 from allele variants from another existing species, right? So, okay, an impressive display of genome engineering. Let's be charitable. Yeah. Say it. Right.
And this is usual in the realm of bullshitters, right? Is that they don't acknowledge limits, right? It's like you get a sample, even how perfect it was, just saying that we now have the full genome of the dire wolf is kind of bullshit. Look, even if you have it, you couldn't.
I love that because that's the technical term, bullshit. I love that. No, that's exactly right, and I couldn't agree.
But then there is also the other part, right? It's like, how much have we actually really learned from doing this exercise, right? Because there are many interesting questions to be answered, and these assholes, another technical term, are kind of burning money on things that they expect to make them more money, but nothing more.
Yeah, no, I couldn't possibly agree more. You'll love my paper on this, where I go in on these issues deeply. I think as you know from philosophy, right, we try to steel man, as we call it. It's a debate technique where you try to be the most charitable possible to the thing that you're criticizing. Right, and you debate that version of it. So what you're seeing is me saying, look, at the very least, fine. You recovered a genome. You were able to use multiplex CRISPR-Cas to introduce these genomes. Cool. But even with that, as I revealed here, you see these kind of unpredictable things at work that actually have complicated the process. So I agree with you completely, right? I agree with you completely. I think we'll continue this. So what I'm going to argue is what we saw there about the unpredictability of putting those dire wolf mutations into a genome is that unpredictably we can actually unpack that. And there might be some concepts here that are kind of relevant for why that didn't work, why the CRISPR companies are failing, and why this entire domain needs to think a little bit more carefully and embrace a complexity science perspective to be able to either do this more responsibly or not do it at all or whatever. I mean, the point is I'm not going to argue one way or the other. I'm going to kind of try to disentangle what's happening here. Plasticity, we'll talk about, performance across environments and contexts. There's a neuroplasticity analogy here, but it's a little bit different. I mean, in some ways not, but how traits that do one thing also do another or multiple other things. This is pleiotropy and then epistasis, how mutation effects aren't the additive sum of the parts. So we'll use those kind of, and I'll talk about work that's gone in my laboratory on each of these three things to kind of have this grander conversation about why the dire wolf exercise is, the technical word, bullshit, and why we need to be more mindful of this sort of technology and approach. And so that's what we're going to do here. So plasticity, the ability of a genotype to produce different phenotypes across environmental contexts. Central idea, again, in evolutionary theory, neurobiology or cognitive science has, it's more neurobiology. It has its own kind of take on this, but I think it's somewhat analogous. This is from a paper I'm writing, and so there's some formal definitions of it, blah, 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 blah. But more than that, I think let's kind of talk about some of its history. I think Sewell Wright, who we introduced earlier, he talked about lability as a condition for evolution. As an organism's got to be able to do different things in different environments. It's kind of like a fundamental part of being a living thing. So this goes back. It's a very, very fundamental, and I think there's some kind of medical examples in sickle cell crisis. It's funny. It's the defining Mendelian disease, meaning the defining simple disease. The one we feel the most confident saying a single mutation actually is responsible for disease, and it is basically your experience with disease is profoundly contingent on environmental things, right? Profoundly. Altitude, diet. People are living longer now than they did. If it was all genes, that shouldn't be true, right? So the point is, even in some of these kind of very simple things that we understand, we use the reaction norm to capture this phenomenon. Like I said, because plasticity is how this genotype is performing across these environments. How this genotype is performing across these environments. Here's the kind of evolution class question I'm going to ask my class in a few weeks. If I say, which one of these is better performing? The answer, of course, is it depends. And that depends is called a gene by environment interaction. Right? Or, right? It's really that simple. When you see crossing reaction norms, that means it depends. What you actually predict for how an organism is going to function is dependent on the environment. So we can do this in a fictional organism. Let's just say we're in Jurassic Park and we're doing disease. These are simulated data. The idea here is you have a wild type, you have a mutant. Let's just say this is body mass, how large it is. And this is small mass, this is large mass. This is the wild type and mutant. What you're seeing here is, right, how large it is profoundly influenced by the temperature in which it develops. It's just a toy example. But we have kind of real examples that we can use. So yeah, talking about dihydrofolate reductase, I already showed you how that kind of, that schematic about how different temperatures influence the enzymatic rate, but we can think about other sorts of things that influence function. And again, let's remind ourselves that we're talking about the influence of mutations, right? So when it comes to treatment, different types and different amount. I don't think there's any chemists here. I was in chemistry when I was on leave at MIT, so I used to have to argue about whether or not these are alike or not alike. I think in this crowd, these are very similar drugs. Okay, they're used at the bedside interchangeably, right? But they're roughly the same size. The idea here is even the subtle chemical difference between these two drugs, which are both antifolates, both dock in the same part of dihydrofolate reductase, have profound implications for the way they work. We can engineer proteins with all of these different mutations. And we can run simulations of how evolution would operate under different drug types and drug amounts. And we can ask questions like this. Maybe a drug A, you have that kind of most likely evolutionary pathway. And drug B, you have that evolutionary pathway. And drug C, you have that evolutionary pathway. So the idea is pharmacological and drug context is influencing how evolution's working. Right? And we actually know this. Even slight differences in drug have profound evolutionary consequences. So again, for drugs that we use interchangeably at the bedside, okay? For drugs that we use interchangeably, which are functionally in the clinical literature the same, right? Let me see this this is summary. This is from an old paper of mine where we actually did this. And what you're seeing is the products of evolution as a function of drug type and concentration. This is one drug, this is another drug. And colors represent kind of a different evolutionary endpoint. So the idea here is even for very similar drugs, the same mutations perform differently. And consequently, evolution takes you down certain routes depending on the drug that you're in, okay? So that's an example of how plasticity is dictating the phenotypic mutation effects across contexts, and more than that, is influencing how evolution happens. So if you're not very, very careful about the sort of environment that you're in, not only is it changing the function of a given enzyme, it's also potentially changing the future evolution, okay? You change things profoundly, and again, if pyrimethamine and cycloguanil are different drugs, I mean, I don't know what the analogy would be for temperature, but in the age of climate change, or whatever I'm supposed to call it now, right? Right? You have to think about how even slight differences in environmental context are going to have consequences here. Next, we'll move on to pleiotropy, the propensity for a single gene to affect multiple, often unrelated phenotypes. This one I love, okay? This one I love. Anybody into Legos? It's kind of cool for grown-ups to be into Legos now. You know what I'm saying? No, you can admit it. We're in a safe space. Come on. You in the Legos. Come on. Admit it. No, I'm just playing. The idea here is, I think part of the thing about engineering, right? And I think that's why it's an interesting word, is the notion that if you think about the way we've talked about biological engineering, even in places like X-Men, it treats genomes like they're Legos. Like you can just move things in and out. Like there's pieces that you just fit together and it's like, oh, this is holding this up, right? But even in there, right, what we're learning is that genomes are not constructed that way. And part of that is because of pleiotropy. Pleiotropy means, if you think about the history of the human genome, and again, the way the history influences this question is very, very important. I don't know how many of y'all were like, I mean, you were all around probably, well, not some of you weren't, during the announcement of the completion of the human genome, 2000, 2001. Some of y'all were around, but it was a very important important time for me. Prior to that, during the Human Genome Project, you remember how many there were? The estimated number of genes in the human genome was supposed to be 200,000. Then it was like, oh no, it's 100,000. Oh, and where we at? 20,000? Why? Or like, why did we get that wrong? I think it's understandable why we got it wrong, because being this successful person takes a lot of biology. I would expect a whole lot of actors to be able to produce this level of excellence. Personally, I would think that. I can't be, there's nothing wrong. You got to metabolize, you got to think, you got to do physics, you got to drink that LaCroix, you got to teach, you got to do all these things. And like naturally, we would think that's composed of a lot of different sorts of biological actors. That makes sense to me. As we begin to sequence, get more information, the number of genes, the actual things that are encoding, and there's some new things about non-coding stuff, blah, 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 blah. There's a lot of song and dance in here. But we have way fewer actors actually doing the heavy lifting biologically than we thought. And one of the ways it's able to do that is because of pleiotropy. Every gene is doing 5, 6 different things. You got genes that function in like cognition, that are functioning in metabolism, that are functioning in locomotion, that are and this is a true thing across the biosphere. So the idea is nature kind of repurposes. It takes things and it says, oh, you have this fold? Cool. Well, now you're going to do this. Rather than kind of necessarily reinventing things or expanding, because genomes are expensive. They're hard to replicate. You can make errors. So basically, and I think the extreme example of this, of course, viruses. You got viruses wreaking havoc with 9 genes. Think about that for a second. We're terrified of these things with like no pieces, right? Relatively speaking. So my point is, this is a kind of a natural thing that we're doing here. And again, there's formalisms, right, that we can use. But again, really thinking about this. So, right, also talked about this. And this is important to keep putting here because like these ideas I didn't invent them. They're old. And I think part of complexity science, it is about new hipster metrics and things like that, but it's also about like deeply reflecting on ideas that have been around. When people thought about them very, very carefully, a mutation, for example, may affect size, color, viability, fertility, and many other characters at once. Darwin, in The Origin of Species, talked about correlated variation as an example of this. And we can talk about this with respect to Marfan syndrome. Abraham Lincoln, who's from nearby, from what I understand. I'm a New Yorker. I don't know, right? Kentucky. But it's autosomal dominant. Again, question.
Or increase the efficiency of the single brain. So is it like a constraint of cost between these two of sorts?
If thinking about it, it's like there's like an energetic cost to having, like, so you're talking about, it's like, the question is, oh my, it's really, really hot here, or oh my, a microbe is like, ooh, there's some really, really yucky drugs here that I'm having a hard time, or there's not a lot of, I need to be able to metabolize a different nutrient. Do I actually integrate a new, do I do a de novo a new gene? Which happens. There's birth and death of a lot of genes, right? Do I do that? Or can I repurpose a gene that I have for something else? Can I tweak some folds? And there's not like a clean answer to like one being preferred to the other, but it's important to keep that constraint in place, and that explains why there aren't as many genes as we think. Because oftentimes nature picks, nah, we're gonna repurpose you. Rather than, no, but also there is the birth of new genes as well. This is why there's so much kind of like conservation. There's a reason why, like, you know what I'm saying, us and like an apple are like 60%. Literally. You are 60% tomato. Okay? Get over it. Right? And a lot of that, or something like that. Somebody looked that up. I think it's 60%. The idea is something ridiculous. But the idea here is it's because, right, innovating isn't necessarily the point. Innovation is in the way they interact. And so nature kind of chooses that as often as it chooses anything else. Right? And so we see examples in the clinical medicine of, again, another Mendelian disease, Marfan syndrome. It's a connective tissue disorder. It has all these different things. But again, look at all the different types of symptoms. That's full body. And so different people have different manifestations of Marfan because the gene responsible does all these things. Right? That's an example of how pleiotropy manifests. Okay? So we can think about this hypothetically again, right? So we have that reaction norm for body mass. What if we did pigmentation? Same mutation. This is hypothetically, just to guarantee that. Same mutation, different traits, and you see different patterns. So the idea is one gene is doing multiple things, and we can think about this, right, in terms of pleiotropy and the de-extinction. Remember that 5 mutations from the direwolf caused defects. What kind of defects did they cause? They were engineered to make it look like a direwolf. And what happened to those extra 5 from the direwolf genome when they put them in the gray wolf genome? Blindness, deafness, all these things that we could not have predicted because those genes are doing multiple things in an organism. So this is a very, very essential challenge for thinking about this. We'll go back to our model molecule to reveal how this works. And this is a paper I love that we wrote. We'll get to epistasis, but we'll focus on pleiotropy, shape biophysical. This is kind of a protein biophysics paper. We published it in Physical Review E. And what we did is we took this fitness landscape, engineered a bunch of mutations into it, and measured a bunch of different phenotypes related to that protein, biophysical phenotypes related to that protein. And bear with me. We looked at, so let's just go down the middle. We did it in 3 different kind of orthologs of DHFR, but let's just go E. coli since everybody knows that. Same set of mutations. Y-axis is kind of how well it performs, you know, some measure of function in that phenotype. IC50 is how resistant it is to drug. KCAT, standard kind of Michaelis-Menten kind of metric for kind of catalytic turnover. This is the inhibition constant. This is the classical Km, which is a property of the active site. And this is the melting temperature. The different topographies of these different fitness landscape is basically saying an individual mutation tweaks these different things differently. See, a mutation that might make you really, 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 really good at catalysis might actually come at a cost. Trade-offs is the most canonical example of pleiotropy. When you tweak one thing and you get lousy at something else, but pleiotropy is much more complicated. It basically says, if I asked you, build me the perfect enzyme, and I want to sell it, how do you do that here when whatever mutation I think is going to take me uphill here might be associated with downhill here? Right? You have multiple things to keep track of when you're trying to engineer, and this is just a single protein. I'm not talking about a complex phenotype like autism or depression or type 2 diabetes, where it's like hundreds of different genes and alleles interacting. So if it's complicated for a simple example, how does that actually map up? All right, right. So, right, yeah, we talked about that. And so pleiotropy suggests mutation effects influence more than one trait. And so the idea that you're going to find a jackpot mutation that's going to get you, right, $1 billion crop, be careful. Right? To recapitulate plasticity, it may not be doing what it does in Illinois as what it does in Iowa. That's plasticity. And then it might help you survive a fungus let's just say it helps you survive fungus in a laboratory in Illinois and Iowa, right? Guess what? It's also making you susceptible to viruses. Right? This is not like my imagination. Like I'm showing you this stuff as the product of the work in my lab and many, many others. So this is another kind of fundamental thing that undermines biological engineering. And then there's epistasis, and this one I love. Maybe one of my favorites. The surprise that the phenotype and mutations are combined given the constituent mutations' individual effects. Okay, I love this. I'm sorry, this is fire. I know what mutil allele X does alone. I know what mutation allele Y does alone. I add mutation X to Y, and I get a phenotype that I could not have easily predicted from the properties of X and Y alone. Seems to this crowd pretty logical, okay? Right? But this completely undermines a lot of fundamental assumptions that exist in population genetics, where we think things are just additive. I've isolated mutation in a gene, right, that does blank, that's responsible for circadian rhythm this or whatever, and I got another one that does this. Oh, I'll put them together, and I'll get double that. And all of a sudden, we see that you have completely nonlinear, non-additive at least, and often nonlinear interactions between these mutations. And we can think about this way. Again, so I thought about this. The effects of gene substitution often depend on the whole genetic system in which it occurs. The reason why I keep putting this is sure, so right is a genius and all that, but like, you're not going to solve, you didn't need a lot of data to think through things carefully. Right? Like the thinking part is important. Think carefully about, right? And we see this in cystic fibrosis. Mendelian trait. You got it. Yeah, it's this very serious thing that we need to take seriously. But there are certain mutations in other genes that actually can worsen symptoms. So the story of cystic fibrosis isn't just about the mutation of cystic fibrosis. It's about cystic fibrosis and the other mutations you have in your genome. It completely changes your experience. And there are other diseases where these things are kind of protective. So the idea is it changes the narrative. I have this joke. I call it a mutation biography. The idea is you got to be able to tell a story about a mutation. It's not just who you are. There's a reason why biographies are long. Because it's hard to tell a simple story about anybody. You have to know where they lived and who they were friends with and all these kind of things like that. Same thing for science, right? So I'm saying this to say we can kind of go back to Jurassic Park. You remember from the story, males were appearing on the island, and who the hell knows, right? But Crichton thought very deeply about a lot of these things and got a lot of things right. They were using frog DNA to complete the gaps. Come on, y'all ain't seen Jurassic Park? That's homework for the trainees. Y'all got to see Jurassic Park, okay? The idea is you use frog DNA to complete gaps in the ancient genome, and those were causing surprising effects, right? And also we saw this kind of in the genome, in the extinction. But we can think about this kind of conceptually here, right? So this is 0, would be the wild type, and you put one mutation in, you put another mutation in, and here, again, you get this pigmentation. You put this mutation, you get this pigmentation. If there's no epistasis, you add those effects and you get here. It's additive. But here you have a positive epistasis, which is a positive deviation from what you would, a positive surprise. Okay, a positive surprise. This is we would call this negative, we would call this sign. Sign epistasis means not only do you not get an additive effect between the two, you get a reverse direction. And so sign epistasis is the one where the surprise is the largest, because you're like, wait a minute, I thought I was getting more of this and more of this, and you put them together, and if you got positive epistasis, you're like, okay, cool, I get more resistance to fungus in a crop. Yeah, I'm okay with that. But what happens if you put these two mutations individually together and you get like more susceptibility to the fungus? This happens, right? This happens, and we see this. So again, we'll use our model molecule again, and we'll talk about kind of this paper we wrote, or I wrote, I guess, about the amount of surprise across contexts. And what we have here is this. Remember pyrimethamine and cycloguanil, the two drugs I don't have to argue with y'all about because this is not a chemistry department, remember? Right? Two similar drugs treated relatively simply. This is the surprise coefficient. We get that. Some of you might be computer scientists from the Walsh-Hadamard. It's basically a variation on Fourier transform. It's been applied to certain sorts of genetic data, small genetic data sets. And it's basically the deviation from additivity. It's a deviation from additivity, okay? So 0, right? So what you're seeing here is, and let's just kind of pick, let's just pick this mutation here, this third site mutation here. What's happening here is, at low drug concentration, not much surprise. You get it in the genome, and the genome, this mutation doesn't have much surprise on average. But the minute you start raising drug concentration, and boom, it has this very, very, very positive surprise. We would call this in population genetics or genetics a compensatory mutation. There are certain environments where it actually is able to drag up the growth rate, right, of this organism. Here's an interesting question. Are these similar or different? Kind of rhetorical, depends on what you mean, right? The idea here is, look, very similar drugs, exact same mutations, slightly, subtly different patterns and surprise across the environments. So the idea here is that epistasis, this nonlinear interaction between mutations, and what this means here is this is the non-additive interaction between this one and the average of these other kind of things that it could be in the presence of. This is a pairwise epistatic interaction between these and the other backgrounds it could be a part of. This we would call this a three-way interaction, right? Or higher order epistasis. And what we're seeing is that surprise is deeply, number one, if I asked you which mutation is the best for growth rate, what would you tell me? It depends here on two things. You actually try to answer it. It depends on two things. It depends on two things. It depends on the other mutations it's interacting with, and it depends also on the environment that it's in. Right? And I'm not when I invented this. This is kind of a real thing. And we saw this kind of play at work here, right? Very, very simple descriptions. This is actually, I like about this by journalist Carl Zimmer. He did a really careful job here. He actually did not put a phenotype. He just kind of put things about it. That's smart. Because he was implying, I don't know exactly what this does, but I do know some patterns about where we find it and what it does. And there's other examples of epistasis epidemiology. We just published this, right? Basically look at how important this is in kind of in viral pathogens of various kinds. And we looked at plant viruses. We basically say how much evidence of epistasis there are off various host species of plant, the gene locus that is interested that we see epistasis in. And basically this is just kind of a systematic analysis to actually show how prevalent it is. And of course in SARS-CoV-2 we see quite a bit. Why are there so many studies of epistasis, evidence of epistasis in Omicron relative to the other strains? Is it because we all of a sudden is it because there's necessarily more epistasis in Omicron than other strains? Or, here's the cynical, that might be true. The cynical view is, by the time we got to Omicron, we knew that simple mutation stories didn't explain. So we actually had to invoke some other analysis tools for that reason. Okay, and we've done this in other sorts of things. So epistasis suggests mutation effects interact in non-additive fashion. So as we kind of begin to land this plane here, right, three concepts that complexify, it's not, the concepts may complexify the act of genome engineering, but they actually help us understand it. They actually help simplify the problems in some ways. That's the kind of the double-edged sword of complexity. It appreciates, right, multiple kind of inputs into a system, but understanding those inputs actually allow you to describe the system much more carefully. Performance across environments and contexts, how traits do one thing also do another, and how everything is an additive sum. And they're not mutually exclusive, and we've actually already seen this. These things actually work in concert. We already saw this. This is plasticity and pleiotropy, right? You have plastic trait across environments, but you have it operating in multiple traits. Right? So we can actually study these things in concert. This actually was epistasis and plasticity. We just talked about this. This mutation that's interacting non-linearly or not additively with other mutations, it's effective that changes across environments. We can actually combine them, right? We have epistasis at work on one trait, has different epistatic effects on another trait across three different environments. So we can combine these things, and maybe we have an inclination, because we're quantitative people, maybe we have an inclination to go, what we need is a deterministic equation that explains it all, right? And that might be useful, right? That might be useful, but I think we need to slow down. That might be useful. And I'm not, I think, we're developing out some kind of mathematic ways to actually describe what a mutation is doing using some of the math of populations, but slow down. I think what we need to return to what the heuristic is, what are the actors, what's the nature of the interactions, and what contexts are modulating that interaction. So when we're talking about soft rules for biological engineering, right, nobody's asked me about soft. Soft rules mean precisely what we're talking about. Perhaps there's a whole area of applied math we can kind of, right, or computer science we can use to properly predict this in a rigorous way so that we can feel confident about a crop or engineering a dinosaur or engineering a dinosaur. Maybe there's a way to do that. But softer rules are, right, we have rules, but we're aware of the sorts of forces that undermine a deterministic picture of how the system works, right? And we can now kind of move forward with that, right? And so remember, right, when we're thinking about this, it's because these different kind of industrial ambitions are having trouble in part because of a lack of appreciation of these sorts of forces. Right? I guarantee the problem with the CRISPR-Cas stuff is that they are dropping mutations in and they are expecting an outcome that's not happening. right? Had they been much more careful about scanning the whole genome, there's practical things you can do here. You can actually identify what the other mutations are. You can take really good granular metadata on the individual patients, and the UK Biobank's doing that. I think there are data there that are actually pretty good at being able to understand particulars about people. But I think we're never going to get this done industrially and clinically until we appreciate this sort of dimensionality. So what do we do going forward? And we'll land the plane here. A context-dependent synthesis in genome engineering, but not just in genome engineering. I think really evolutionary theory needs it much more rigorously, right? The idea is an appreciation for these things, but again, these are the things that I like and think about and work on, and ultimately I think are relevant. Importantly, this is not just about genomics, right? Is there an analog for plasticity in robotics? Yep. Is there an analog for pleiotropy in robotics? Yep. How about in microbiome construction? Yep. Virtually every type of system that we're trying to construct has an analog to these sorts of things. So I think we might be actually potentially can construct a broader theory of complexity in engineering, right? There's a language at SFI called emergent engineering, where it thinks about how to think about systems this kind of way, right? And so that's kind of one of the things. But it's not just that. There's other things to think about and how they may modulate our understanding of complex systems, robustness, noise, and stochasticity. Somebody here got to do that. Professor, you're a statistical physicist, and they just go ham on this stochastic noise thing, right? In a good way. Epigenetics, we're not going to go there, but that's all. It is an important player. We can in the Q&A, right? And the profound role of culture in shaping genotype-phenotype maps, right? And culture meaning a lot of different things, and I mean human culture, but it's like behavior as a shaper of the way genotype-phenotype maps work. Right? What's the role? Yeah, how do we know what the dire wolves? Even if you could de-extinct the dire wolf for real, how are they going to operate in a world without other dire wolves? That's kind of a deeply important dimension to the manner, right, that exercise will succeed at all. So thank you for your time. I'll take some questions and discussion points here. Some of this is from this preprint is out, but the paper's in press. And a very, very provocative paper I'm writing called "Can an Engineer Fix a Genome?" which of course is a play on can a biologist fix a clock radio, which is a famous paper. All right. And we got a few minutes.
Thank you. Thanks for the great talk, Brandon. I have some questions, but why don't I first give the floor to others if anyone has anything they want to ask, or I can get the ball rolling.
Great talk. So obviously you hinted at these principles of pleiotropy and epistasis can be applied across a number of technological systems. To what extent do you see these factors playing around in human systems and behavioral systems? I'd love to hear you talk about
yes.
that.
So when I introduce epistasis as a joke, I have this figure where I say, I have one student, when they team up in computational biology, they do beautiful. I get this beautiful additive effect. When they team up in experiments, I get this positive epistatic effect. And when it's time to write the paper, they get a negative effect. Nobody wants to write the paper, right? Meaning in many ways, so yes. So there's a lot there with regards to human behavior and the way you stick things together, right? In fact, one of the best systems, and this is great for this community, one of the best systems to think about this sort of things is sport. I mean, like it's the best data on human behavior ever constructed. Think about basketball. There's 5 loci on the court at a time. We have data on how individuals get swapped in and out. Why is it that, right? We have these trials from the Olympics in particular, where countries who played together longer have much more positive epistasis. The 2004 USA team, which had the best talent, lost. Why? Because you had antagonistic epistasis between the players. I mean, we can actually demonstrate this quantitatively. So I'm just giving you a kind of a popular culture iteration, but that probably applies to everything. It applies to why NICO, is that what it's called? NICO works so well. That's what you have to do. You have to curate. All team formation is curation of individuals and hoping you get a positive effect, at least additive, but hopefully even positive epistatic. That's what curation is of any social organization. So a lot more there, but yes. Well, one of the things one doesn't want to do is like simply take the actual map, like take it and just walk it and airdrop it into another system. You must understand some features of that system. So this is why domain is, for the trainees, domain knowledge is important for that reason, right? So you actually know what you're talking about and can explain it. So I think that's very, very important. I think thinking about this with regard, knowing kind of how context dependent your system is, using the sporting example again, is the system you're operating a baseball style system where it's basically an individual sport masquerading as a team sport? That's really what it is. It's individual interactions that we can measure between pitcher and batter. It's much less epistatic than soccer or basketball, and football is a disaster. American football is a complete disaster. So the idea is, this is actually something I want to encode systems as different sorts of systems. And once you have that information, yeah, you can apply the math to it. You can apply these computational tools to it, but you have a much more of a you're not going to say anything stupid because you understand the context of the sort of sport. So I'm developing a taxonomy for complex biological systems that are baseball style systems versus football style systems. Football has a lot of nonlinear interactions, but they also have a quarterback. So that's an individual actor that actually does have a profound influence over the performance of the system, but everything else is deeply context dependent. Isn't that weird? Yeah, I got some cricket people around me. I got a cricket in my lab, so I'm trying to learn. Apparently games take 4 days and all that, so I haven't done that yet, but we're getting there. Apparently cricket is this times 1,000. So yeah, great question. Sorry.
Just because it's relevant, I want to throw my question in here.
Please.
To these last two questions.
Mm-hmm.
What do you see as the solution to like the astronomical number
Yes.
of possible interactions that arise? Not even when you have 5 people on a basketball team, but when you go to the number of genes.
Beautiful, beautiful question. So let me turn the volume up on the cynicism there, because it's one I ask myself, right? Or let me ask a different question. Doesn't epistasis feel a little nihilistic? Like there's no way you could ever understand any system ever in the universe because there's too many interacting things. You have to understand the way they all interact. And if you have like 1,000 genes, you would actually need to think about how they all interact, right? So that's like a criticism. It's not a formal criticism of that paradigm, but it is a real one, right? Do you really need to understand everything about everything? And the answer is no. There's a paradigm called global epistasis that I didn't talk about. And what global epistasis basically says is that there's actually a mathematical function that explains the deviation from additivity. It underlies a lot of systems. And we published on this. And so the idea here is there's something about ensemble. It's a property of, it's a physics thing. It's a property of ensemble systems that there's still an underlying rhyme and reason that can predict the deviation from additivity. And I think that's probably a feature of a lot of these systems. Does that make sense? Yeah. Yeah.
I feel ashamed because I always talk too much and ask too many
No.
questions. So the first thing is I want to thank you for an outstanding talk and for also doing something that I think is very brave and that the bullshitters don't do, which is tell us that reality is complex and it's hard. And I don't see it as nihilist. I think that it's good to acknowledge that these things are difficult and we are not going to solve all problems. And one of the big issues that I find with pretty much everything, science, politics, policy, all of those, is people like the simplistic approach. There is one thing, I do this, the problem gets solved, right? The bad apples in the police, the bad apples on ice. If only we can just do these things, everything will be solved, instead of looking at the system configuration and what's going on. So yeah, thank you very much. I love this talk and I really appreciate that you are telling people that the simplistic approach may be attractive, but it's going to lead you astray. And so it's good to acknowledge the interactions, the many parts and the system and context things.
I like you. Can we be friends? I like this dude. No, really, thank you. No, thank you for that. And what I loved about that is I didn't say that loud, that message was kind of embedded in what I was saying. I didn't tell you, things ain't simple. I did say it a few times, but I didn't, that wasn't the gospel. I did this through a biological problem, but I think this is absolutely what my gospel is. We shouldn't be afraid of complexity, and complexity does not imply nihilism. There are rules, but maybe they're soft rules and not hard rules. And I think this is what I try to apply across all my systems inquiry. So thank you for landing on that.
Stopping point. We're at time. So thank you very much again, Brandon. Let's thank him
Yes.
again. And
Appreciate it.
thanks for
Thank
coming
you
as well.
Great questions.